For the August 31st in-class Socrative quiz, here are the answers. First, for the given DNA sequence, we need the complementary strand. So the concepts here, the base pairing, that T's will always be opposite A's and vice versa. G's will always be opposite C's and vice versa. So that takes care of base pairing and complementarity. So given TAGACAT from the 5' to the 3' ends of that molecule, the complementary nucleotides would be T pairs with A pairs with T, and so on. And then we need these two strands of DNA. We'll call them strand A and B to be anti-parallel. That means that the 3' end of the top strand is going to be next to the 5' end of the bottom strand. And likewise, the 5' end of the top strand is base paired with this T and this A are base paired together. The 5' end is next to a 3' end. That makes these two strands having anti-parallel orientation. For question 2, the genome has to have 100% A plus T plus G plus C. And if the genome contains 15% A, and if every A in the genome is paired with a T, that means that the genome also has to have 15% T. That means 30% of the genome is A plus T, which means that 70% of the genome, the rest, has to be G's and C's, and those have to be divided evenly because of complementation and base pairing. Every G is paired with every C, so it must be 35% G and 35% C. So if we come back and look at the options, we can tell that the percent G needs to be 35%. Question 3 asks about the polarity of this image of DNA. Do we read it from the top to the bottom? So we need to determine the orientation of this molecule. To do this, first let's write down the carbon numbering. Here's the nucleotide base. It's always connected to the 1' carbon. And then there's the 2' carbon. This is the carbon that in ribonucleic acid would have an OH group here. This is deoxy ribonucleic acid, so it has no OH group there. 3' carbon, 4' carbon, and there's the 5' carbon where the phosphate is attached. Here at the end of the molecule, we have the 1', 2', and there's the 3' end with the free hydroxyl group. So the 5' end is defined by the presence of a free phosphate group, the 3' end defined by the presence of a free hydroxyl group. So this is the 5' end, this is the 3' end. So we read this bases in order. Here's base A, the second one, base B, base C, and base D. And so we should read this nucleotide sequence from top to bottom, which is from the 5' to the 3' direction. Now, I already mentioned in the answer to the previous question, this molecule is DNA because at the 2' carbon there is no OH. And as one of you pointed out in class yesterday, the other reason that we know this is DNA and not RNA is that one of those nucleotides is thymine, which is only found in DNA. Uracil would be found in RNA. In question 5, we learn that the DNA sequence here is actually CGAT, which means the opposite strand will be GCTA. And if this is 5' to 3', the opposite strand would be antiparallel, 5' on the right, 3' on the left. So this question is asking how many hydrogen bonds will be made between the strand that's shown and the antiparallel strand. G's and C's each make 3 hydrogen bonds with each other, and A's and T's each make 2. So if we total those, there are 10 hydrogen bonds between these 2 strands of DNA. Likewise, if you have one strand of DNA that has C's and G's and one that does not, it's pretty easy to tell that the strand with C's and G's will have more hydrogen bonds than the strand that only has A's and T's. Therefore, strand A will have its strands held, or double-stranded molecule CGAT paired with its complement will have more hydrogen bonds than TTTT, which would be paired with 4 A's. There would only be 8 hydrogen bonds in that double-stranded molecule.